Today about some of our work on neural codes for natural behaviors in flying bats. And so I'm generally interested in the neural basis of behavior, and I often ask myself, which kinds of behaviors should we study? So for example, if you're interested in social behaviors, then you may want to study something like this. Group hunting in lions and communal defense in buffalo are really very interesting behaviors. But then you might say, maybe not very controlled, not very reproducible, so hard to study. So then what most neuroscientists at least do is, instead of don't focus on these kinds of natural behavior, but studying something more close to this. This is a tube test in mice if you want to determine their hierarchy. So you put them in a tube, and basically the one who pushes the other out is considered the winner, so sort of the higher in the hierarchy. And so we have this, I think many of us, certainly in this room, have this notion that there is this axis between, on the one hand, control of order experiments on the left that are controlled but unnatural, and on the other hand, natural behaviors on the right that are natural but uncontrolled. But what I'd like to argue today that this is actually not so simple, because, for example, the lions and the buffalo, it's not totally uncontrolled. I mean, the lions are not randomly running around. And in fact, people have studied in quite detail, and they have repeated behaviors, and they do things in somewhat systematic ways. It's not totally controlled, but it definitely has some amount of control that the animal is doing for us. Control meaning repeatability. So we have hope to understand what's going on. On the other hand, the tube test is also not completely unnatural. I mean, these are burrowing animals. They do meet each other underground, so it's not totally unnatural. So I would like to propose, in fact, it's more of a kind of two-dimensional axis. In a schematic way. So we have the artificial to natural axis and the uncontrolled to controlled. So of course you can have extremes. So let's choose a very artificial and highly controlled behavior that has been studied in neuroscience a lot, head fixed kind of cell stimulation. Okay, that's clear. Highly artificial, highly controlled, and the other extreme would be 24/7 unconstrained behavior. So that's highly natural but highly uncontrolled. And so this is sort of the line connecting them is sort of the this axis that I showed before. But then you have things that are totally off diagonal. For example, birdsong, one of my favorite examples, and I think we'll hear later. So the bird is repeating, or some of the birds are repeating the same song, or with a millisecond precision, or a few millisecond precision over and over, tens or hundreds of times a day. So that's extremely controlled. It's actually more precise than a lot of the behaviors that we strive to achieve in the lab. So you can have behaviors that are both natural and controlled, and the lion hunting might be somewhere here, highly natural, but maybe a bit less controlled. And so I'd like to argue that this is the conceptual region we need to work at. Behaviors that are natural, but at least offer some amount of control, in the sense that the animal is repeating its behavior for us. Okay, so I'm studying mostly the neural base of navigation, and so this is sort of where we'd like to get, and I don't think we're quite there yet, but basically today I'll tell you about our journey towards this sweet spot. So what do we know about the neural base of navigation? So this is now 60 seconds about the cell types that you all have heard of. So placed in the hippocampus, there are place cells. If you let a rat run in a maybe 1 by 1 meter empty box and photograph it from above, then you'll see the black line here is the trajectory of the rat, and the red dots are spikes. So this particular neuron is activated in the northeast corner. So this is the place cell discovered by O'Keefe in the 70s. Then in the medial terminal cortex, there are grid cells discovered by the Mosers. The experiment is basically the same. There's no navigation task here, really. You just randomly throw food pellets in this 1 meter box, but these neurons fire in a set of positions that form a hexagonal lattice. And then there are also neurons that don't care so much about the position in space, but represent the direction in space. Okay, so we have the place cells can be thought of as mental maps or cognitive maps of where am I in space. Grid cells can be perhaps used to measure distances, so rulers, if you will, or also to represent position. And we have neural compasses, the direction cells. So this is sort of the elements that you'd like to build a navigation system in the brain. Now, what we ask in my lab is, how does real-life navigation differ from navigating in a 1 by 1 meter empty box? Because most of the studies in the field are indeed when the animal is navigating in small boxes or maybe on a short linear track. And you can give several answers. One is that the world is not flat, it is complex and three-dimensional, so how does that affect the system? The world is not empty, it contains goals. How do we navigate to goals? The world is not 1 meter in size. Animals and humans navigate kilometers. So how does scale affect the system? And also many animals navigate in groups, and so how do social interactions affect the system? And you can give more answers here, but these are things that, that we address directly experimentally in my lab. And in order to keep in time, I'll just focus mostly on the last two. I'll mention a little bit the first one today. So now we studied this not in rats, we studied these questions in bats. Egyptian fruit bats in particular, you see them here, very large bats. And it's important for me to say that the idea is not to understand the bat per se, but to understand the brain. But as I hope to demonstrate today, by using this this interesting animal, we can utilize its interesting behaviors in order to ask questions that are difficult to answer in rodents or in humans, for that matter. So to put you in the mindset of what does it mean to navigate for a bat, this is from a study we've done some years ago where we put GPS data loggers on bats outdoors. Okay, so this is a purely behavioral study outdoors. We just put little GPS data loggers to track their navigation outdoors. And here's an example from one animal that flew from its cave to the same individual tree night after night. So each color here is a different night for the same animal. So you see that this animal came back to the same tree night after night and flew in a nice flyway. I mean, there is some jitter there. It might fly a little bit to the left or to the right, but it definitely knows where it's going. They're great navigators. And so these are the kinds of behaviors that we'd like eventually to understand neurally. I don't think we are quite there yet, but we are heading in this direction. So I'll tell you very briefly about what we found in 3D representations in the brain of the bats. First, before that, just to tell you, in 2 dimensions, when we sort of ratified the bat and let them on purpose crawl in a 2-dimensional box, just like the rodents, we found the same kinds of neurons, place cells, grid cells, and head erection cells. In 3D, Michael Yartze, when he was a PhD student in the lab, had the bats fly in a large flight room, 5 by 6 by 3 meters or so, and he found place cells that look like that, so that have spherical place fields, spherical receptive fields, if you will, and that were about maybe 80 by 80 by 80 centimeters. And the fact that they're spherical means that the resolution, at least in this flying mammal, is the same in the X, Y, and Z dimension, the resolution of the representation. Then a couple of years later, Arseny Finkelstein looked at are there 3D head erection cells, 3D compasses, if you will, and indeed he found neurons that represent a particular 3-dimensional direction in space. And just to say that following these studies, several other groups started to look at these questions at monkeys in particular, and recently also in mice, and found essentially that there are 3D head erection cells both in monkeys and mice that share a lot of the properties for the bats, and recently also 3D place cells in rats that move in this kind of 3-dimensional volumetric maze, quite similar to what we found. So it's certainly not just in bats. Now, what about grid cells? I'll just mention very briefly because I'm giving a full talk about this tomorrow in the grid cell workshop. But just to, okay, these are the kind of the classical grid cells from rodents from the Moser lab, and when Gili Ginosar, a PhD student in the lab, recorded grid cells in the entorhinal cortex of bats that were flying in the same flight room, she found neurons that looked like that, and there was a fundamental difference from classical grid cells. So she found that there are fixed local distances or a local metric of space, meaning that these fields tend to have fixed distances between each other, okay, locally. But there was no global lattice, okay, no FCC or HCP, no hexagonal 3-dimensional lattice. And if you want to hear more details, welcome to the grid cell workshop tomorrow at 7 p.m. I'm going to give a full talk just about that. Just to give you the intuition for if you're now confused about how can you have fixed local distances but no global lattice, just to show you that there is a fundamental difference that we know already in physics for many years between 2D and 3D. So if you take spheres and jam them together, so like on a two-dimensional surface, like on a pool table, you jam them together, so you'll get this hexagonal arrangement. But if you put a lot of spheres into a three-dimensional box, like in a gibbery pool of balls, you'll never get a perfect lattice. You'll always get stuck in some local energy minimum. However, if you look in this ball, then its distance from its local neighbors is fixed. It's actually exactly the diameter of these balls in this case. Okay, so this shows that there's a fundamental difference between 2D, where it's easy to get these hexagonal structures, and 3D, where you can have a local structure but not a global one. And we had another model, not with balls, but a different one inspired from physics. But basically, this model led us to kind of explain both what happens in 2D and in 3D, and therefore we suggest that the fundamental property of grid cells is not the hexagonality per se, but having multiple fields with a characteristic pairwise distance between them, and if you restrict them to be in a two-dimensional surface, then you get a perfect hexagonal lattice. But if they're in 3D, then you get a local metric space or a local distances, but not a global lattice. And the implication of this, I think, is very important, because there's a lot of models of grid cells that rely on this precise periodicity for representing space, for navigation. There are all sorts of models out there. So basically, any model that relies fundamentally on having a precise grid will not work, at least in 3D. Okay, so for more details about the model and the results, you're welcome to hear more tomorrow. But this is just to show that by looking at another animal, by going from 2D to 3D, you can find quite different results. All right, now I'll tell in a little bit more detail about two studies. One about social interactions, so social place cells and social time cells in the bat hippocampus, and the other one is long distance, representation of long distances. So I'll start with first the social side. This is work by David Omer, a former postdoc who just started his own lab at the Hebrew University. So basically the question that David asked was the following. Bats are highly social mammals. So he asked, in the hippocampus, where there are neurons that represent where I am in space, okay, the place cells, can you also find in these animals neurons that represent where you are in space, so the positions of other individuals in a social context? So to address this, we designed this task. We call it a delayed match to place task. Looks simple, but actually not so simple. So it involves two bats, a demonstrator bat in blue and an observer bat in red, which is also implanted in the hippocampus. So the demonstrator bat's task is to fly from the start ball to ball A or to ball B, pretty randomly. So in this case, let's say it flies to ball A and then back. And then after a delay, the observer bat in red has to mimic it. So in this case, has to fly to ball A. Okay? If the blue bat flies to ball B, then the red bat also has to fly to ball B. Okay? So it's a mimicry task. But it has two key features for us. One is attention. The implanted bat has to attend to the location of the other bat. So we have hope to find representation of the other bat. And the other, you noticed in this cartoon that one bat was flying coming back, then was a delay, and then the other one. So they were not flying together, and we did this on purpose because this is the hippocampus. We have place cells. So if there were two flying together simultaneously, it will be tricky to disentangle the representation of myself, the place cells, from the representation of the other. So we built it this way, such that when the other one is flying, I'm not moving, and just to be sure, we had an accelerometer on the head of the bat to rule out any miniature head movements that can explain the results. So I won't show you these controls, but they indeed cannot explain the results. So responses really are to the movement of the other bat. And so this is an example of one neuron that David recorded in C1. So this is all one neuron. What you see on the left is the self movements. Okay, these trajectories, the gray trajectories, when I'm flying, and you see that there's no red dots here, no spikes at all. So this neuron does not emit at all any spikes when it's flying. So it's not a place cell. definitely not place cell. On the other hand, when I'm not moving anywhere, but you're flying, on the right, these are trajectories, your trajectories, then you see that this neuron emits spikes, in this case, on the right-hand side. So representing the position of the other animal, so we call these neurons social place cells. Of course, there are also classical place cells. This is an example of a neuron that fires in a particular location for me, but does not respond to the other bat. Okay, so classical place cells. And then there are neurons that are doing both. In this case, a neuron that fires for me on the left-hand side and for you on the right-hand side. So in this case, the kind of position or the receptive field for me and for you are in different locations in the room. In some other cases, they were overlapping. There's clearly a representation of the position of the other bat. And again, this is already published. I'm telling this part of the story quickly, but we had various controls. For example, you could say maybe it's not really representing the position of the other bat, but maybe while the other bat is flying, I'm planning my own movement. So these are kind of planning signals. So we have 3 controls to rule that out. I won't go into the details. And also, maybe it's not really social. Maybe anything that moves out there through a response field, if you will activate the neurons. So we had also experiments where we had plastic objects move through space, and interestingly, some of the neurons responded to these plastic objects, but the responses were always different than to the live animals. So it seems that there's really something special and interesting about this live conspecific. So these are really social responses, and so we think that these social place cells may be important for social spatial cognition, representing conspecifics, predators, prey, things like that. I mean, we are all as humans, we're good in knowing the positions of other individuals. I have kids, so when they were little, we go to the park, I always have to pay attention where they are, or where's your spouse or your friends, or what have you. So we're pretty good at this. So maybe these neurons are related to these capacities. Okay, so this was about the social place cells. Now I want to tell you in a bit more detail about time cells. This is unpublished work. Also by David Omer, basically from the same experiment. So what I'm going to tell you is reanalysis of data from the same experiment, but just looking at things from a different angle. So first of all, what are time cells? I'm sure some of you know about them, but just let me explain. So these were discovered by Pastelkova and Buzaki in 2008. Here Buzaki doesn't like calling them time cells. Howard Eichenbaum called them time cells. But in any case, the experiment is pretty straightforward. So you have a rat running on a kind of figure-8 maze. So this is a classical task, kind of an alternation task, so kind of to alternate directions. And what they introduced here, the difference that they introduced in this particular experiment was they introduced a running wheel. So before the rat runs on the stem of this figure-8, it has to run for 15 seconds on the running wheel, sort of not going anywhere, running in one location. And then what they found is that there were neurons in C1, in the hippocampus, that fired in particular moments in time. So this is now 30 neurons over these 15 seconds. So this shows you sort of the PSTHs, or the firing times of these individual neurons. And you see that each neuron fires in a particular moment in time, forming these sort of sequences. So these are called time cells. Now what we wanted to address here is 2 questions that were not addressed so far. One is simultaneous encoding of time and place, or space-time, if you will. And you see where I'm going, because in the experiment I just told you about the social place cells, we had 3 positions in the room: the start ball, ball A, and ball B. So where the animal, for part of the time, the animal was hanging and doing nothing. And so you can ask, are there place cells, and are they different between these different locations? Of course, you can do this also in rodents. All you need to do is to move this running wheel to 3 different locations. This is never done. Okay, so this is one question. Are the neurons represent together time and space? And also time cells in a social context. So this is what we'd like to address. And so first of all, yes, there are time cells in bats. So what you see here is an example neuron, and this is the raster. Time 0 here is the landing of the bat on one of the balls. Okay, so it lands and then it hangs there doing nothing, and we can make sure it's doing nothing because we have the accelerometer, the same accelerometer, so we can rule out any movements. And you see that this neuron fires at a particular moment in time, maybe 2 seconds after it landed, almost every time. Okay, these are 32 landings, 32 trials. And here are some more examples of neurons that fired early, neurons that fired later. And you'll note that cells activated at a particular time despite wide variation tri-duration. We didn't impose any trial duration. I mean, the experiment was designed for a different reason. But the animals decided when to take off. And you see here that we ordered their trials by their duration. You see that there was a very large difference in the trial duration. Still, the neuron fires at a particular moment in time. So it really seems to represent time from the landing, irrespective of the kind of future duration of the trial. And we had accelerometer data that we could use to rule out the possibility that these are movement-related responses. Again, I'm not going to show you these controls, but these are really internally, this is an internally generated activity. The animal is really hanging, doing nothing, and these neurons are activated in particular moments in time. And this is we now order all the neurons on ball A and ball B on the start ball. This is how it looks. Okay, so we have sequences of firing. So this is all basically reproducing what has been done already in rodents. Now we can get to the fun stuff, which wasn't done before, and that is look at the representation of time and space together. So here what you see are two neurons, okay, these two rows, when the bat was landing on the three balls, ball A, the ball B, and the start ball. And you see that this neuron is a nice time cell on ball A, but doesn't fire at all on B or on start ball. But this neuron fires on ball B, but not on A and not on start. So really these neurons, you can decode from them, and we've done decoding, I'm not showing you that. You can decode simultaneously time and position in the room. You can look at it more formally by taking all the neurons that are only significantly tuned, significant time cells on ball A, ordering them by the preferred time and plotting the responses of the same neurons on ball B, and you see that the kind of this breaks down, and vice versa. If you look at the neurons that are tuned on ball B, they have no order on ball A. So this means that really the neurons have distinct representation on the two balls, so they represent time and place, if you will. We can call them episodic cells. There was also an interesting subpopulation of neurons that fire on the two balls together, on ball A and B together, that have significant responses on ball A and B. And if we plot them, the neurons on ball A, and we plot the response of the same neurons in the same order on ball B, you see that there is kind of this halo that looks similar to the original order. And indeed, if we plot the time difference between the firing on the two balls, this is kind of the pink histogram. The time differences are very small. But that means that there's a subpopulation of neurons that are pure time cells. If you fire on both ball A and on ball B, then you tend to have the same preferred time. And I'm not going to show you this, but these are really two distinct populations. You can plot the histogram of this correlation between A and B, and this is really a bimodal distribution. So there's two different populations, one that represents time and place simultaneously, and the other one that are these pure time cells that have the same preferred time on the two locations. We also looked at the responses to the other bat. We can do this same trick of time, what happens after the other bat has landed, and there are neurons that fire in particular moments in time after the other bat has landed as well. So neurons representing time for another animal. Okay, so there's really a very rich representation for both space and time for another animal. And we are now running an experiment by my postdoc Psychiat Ray, really the most crazy and maybe uncontrolled experiment, if you will, where Psychiat has set up this colony room, so 6 to 10 bats together that live 24/7 in this colony room, and we developed methods to track the identities, positions, and head directions of 10 animals simultaneously in a three-dimensional complex room. Okay, this one sentence I just said, it's 1 year of work, and it's not trivial at all. But we have that working, and so now we can record neurons and ask all sorts of questions. For example, is there a different responses to different individual animals, right? So far I just showed there was a response to one bat, but like are there different responses to different individuals, kind of Jennifer Aniston bat cells, if you will. Males versus females, because now we have here males and females, dominance hierarchy. Then we have really interesting, we have here kind of two tracking systems, both a system of 6 cameras, so it's really like a big brother. We see all the time what's going on, and also this RF-based localization, two-dimensional localization system. And we see there now with all these cameras, we see behaviors we've never seen before. For example, some of them we have no idea what they mean, but like so there's the behavior of one bat is, first of all, there's food sharing, which I don't think exists so much in rodents, but where bats willingly share food with each other, very interesting. And also another behavior where a bat is hanging and then another bat is coming and sort of landing on top of it. Now it can land anywhere it wants. It's a free ceiling. It doesn't have to land on top of its friend, but it's sort of like almost as if they're playing. And the other bat is doing the same thing, but it's only these two bats are doing this. Bat A is landing on bat B, B on A, but they don't do these other things. So there's clearly very kind of reciprocal and specific behaviors, and we can then try to look at whether neurons represent all of my friends, the ones that I like to interact with. So a lot of questions that we are starting to ask here, and kind of coming back to what I said in the very beginning, you'd say this is hopeless, right? They can do whatever they want. There's no task here. They're living 24/7, and this will never work. Maybe you're right. We have to prove that it's working, but the good news for us is that they are repeating particular behaviors over and over. They're fighting over and over. They're food sharing. The sex. They're doing a lot of things over and over. So you can really, you can build a raster. If you record for 3 hours, you can have enough repeats of a particular behavior that we can really try to understand what's going on, and we already start seeing really interesting stuff. I hope in a year we'll really have some interesting story here. All right. Let me switch gear and tell you about the last story that I wanted to talk about today, and this is the representation of large-scale spaces in the brain. So this is work by Tamir Eliav, a PhD student in the lab. So bats really navigate both underground, hundreds of meters, really long distances underground, as you see in this cave system in Israel. By the way, it's a human-made cave system, so it's really an old one. It's really interesting places to visit. And also outdoors, as I just showed you. And this really serves to illustrate the problem. Okay, if you take this flyway, this is just an example, this particular flyway, the volume that the bat navigates through is about, in this case, it's 15 kilometer long, 2 kilometer wide, and about half a kilometer high. Okay, so they're flying from about 100 meters altitude to 600 meter altitude. So we're talking about a volume of 15 kilometer by 2 kilometer by half a kilometer. Now, if you'd like to tile this volume with your laboratory place fields that you measure in boxes, which are about 10 centimeter in size, then you're in Cosyne, you can all do it in your head, don't even need a back of an envelope. So you need about 10 to the 13 neurons or so, roughly speaking. Now, entire dorsal CO1 is about 100,000 neurons. Okay, so we are, depending on assumptions, you're between 8 and 10 orders of magnitude off. Okay, there's no way this is going to work, just impossible. There's no way that we found in small laboratory environments, in small boxes, translates one-to-one to what's going out outdoors. There has to be something else. And so what is this something else is what Tamir was after. So to address this, we had to solve three technical problems. One is that we have to be able to record neural activity at essentially unlimited distances. The early recordings that we did with the place cells, they were wireless, but they would transmit the data. This could work a few tens of meters. This will not work over really long distances. So we, together with Jacob Wecht, a great engineer, developed this neural logger device that gets smaller and smaller. This is generation 0. Now we're generation 4. It's much smaller. And this really stores the data on board the animal. So now we can record essentially at unlimited distances. And you can get really nice spikes. Here are the 4 channels of one tetrode recorded in flight. You can get really nice spikes in flight. And by the way, in the last few months we switched to a kind of 16 tetrode microdrive and a 64 channel neural logger that also has kind of ultrasonic microphone on board and a 9-axis motion sensor. This is kind of an entire onboard flying EFIS lab. So kind of we're trying to move everything on top of the bat. Potentially we put cameras and a lot more, all very, very small. The second thing we have to solve is how do we measure the position of the animal precisely? You'd say use GPS. GPS is not good enough. GPS is about 10-meter precision. The reason is that you measure time differences, satellites in outer space, and you have atmospheric aberrations. And we found this solution from a French company that has something that's a bit similar to GPS, but you kind of deploy ground-based RF antennas, these green antennas, and then you can measure the distances from the antennas, these circles, or spheres actually, really, and then you can measure the position of the animal very precisely, and we get to about 10 centimeter precision over long distances, actually even better than 10 centimeters, about 8 or 9 centimeters, so very precise measurement of the animal's position. And lastly, we want a controlled but large behavioral setup. So we built this tunnel 200 meters long. It's just the end of it is actually much longer than this, and we put landmarks inside. And then the bats kind of really love flying there. This is kind of time within a session, and this is 200 meters of position, and you put food at both ends, but the bat is flying back and forth and back and forth and back and forth. Many of the bats don't even care about the food; they just enjoy. I mean, this was the worry, right? We're going to build this infrastructure, and then if the bats, we're going to release the bat, and the bat is going to not want to fly. What are we going to do? A problem. The first time we release a bat there, all my worries evaporate. They just love flying. These are marathon flyers. We keep them in nice flight cages, but still few meters, small cages, and suddenly we release them in this essentially infinite or a very large space, and they do what they love doing, flying back and forth. The good flyers are flying 100 or even 120 laps in one recording session. We're talking about 20-25 kilometers of flight in one recording session. It's pretty crazy. And they're flying. This is a cross section of the tunnel. We built it kind of this V-shaped or inverted V-shape, kind of funnels them to fly in the middle. So you see that they tend to fly in the middle, and also not much of a span in the Z-axis. So this is really kind of a one-dimensional behavior. So if there's all the jokes about a one-dimensional cow or one-dimensional horse, so this is as close to it as it gets with behavior, really, and we want it to be one-dimensional, so we can measure very precisely the representation over long distances. And the velocity of the bats is very high, in this case 7 or 8 meters per second, sometimes even 10 meters per second, and you'll notice the behavior, the flight velocity or flight speed is extremely constant across the entire tunnel. This will be important in a second. So here we are essentially utilizing the bat's flight speed to measure the representation of large spaces, right? Because it flies at almost 10 meters per second, we are able to get these many repeats. If you'll put a smaller animal or a ground-based animal, it will probably do a couple of runs and that's it. And I'm done for today. So here we're utilizing just the flight speed of the bat to ask questions about large spaces. Okay, so this is an example of a neuron that Tamir recorded in dorsal CA1. Of a bat flying in this tunnel with everything on it. So all of this is one neuron, and here are rasters. We separated the rasters to the two flight directions, the blue direction to the east and the red direction to the west. And this is a raster, so each line is one flight, and each dot is a spike. You see several things here. First of all, that there's a difference between the blue direction and the red direction, okay? And that's actually old news. We know also in small environments that place cells fire differently in two directions on a linear track. The more interesting things is, first of all, there are not one place field, but many place fields. So we see that this neuron has many receptive fields. Some of these receptive fields are very large. You see here the receptive field is huge, it's 20 or 30 meters in size, okay? This is dorsal C1, where you're supposed to have 10 centimeter place field. So certainly when you increase the environment, things scale up. And the most surprising things for us was that what we call a multi-scale property of these neurons, that the same neuron can have different resolutions in different locations, meaning it has a large place field here and a small place field over there. Here's another example neuron, a small place field here, a small place field here, and a medium place field there. Okay, and we have now recorded a couple hundred neurons. It's very consistent that we have these kind of large. We have large and also small fields. The smallest fields are 60 or 70 centimeters. Okay, so despite the fast flight speed of the bat, there can be very, very small place field, but also very large ones. Each neuron has multiple fields, and this multi-scale property looks very, very different from place cells recorded in small laboratory setups. Just to show you some population results, this is the number of number of cells that has a particular number of fields. So you see that some cells has 10 or 20 place fields, receptive fields per direction. Okay, so it's really not this notion that we have of a Gaussian with a single receptive field, but you have multiple locations. And also this multi-scale property. So this is the ratio of the largest divided by the smallest field for the same neuron. And you see that some neurons, the ratio is 10 and even 20. 20 means that I have the same neuron has a place field that's, let's say, 1 meter here and 20 meters over there. So very different resolutions for the same neuron in different locations. Very, very different than our notions on place cells. And we had some controls that are going to skip over. Fields do not, if you're worried that maybe this is caused by by the landmarks, so it's not, fields do not cluster near landmarks, in particular small fields don't cluster near landmarks. You could say maybe flight speed can explain it, maybe when you fly faster the fields are larger, but no, you've seen that the flight speed is actually extremely constant. Variations are a few percent, but the difference here can be 20 or 10-fold, so it's definitely, we can show this doesn't explain it. Spike sorting is, we're always careful with spike sorting. Here we're ultra careful because this is like your first worry is that these are just you're mixing up neurons. So this is definitely not the case, and we can show that. Okay, so a lot of things we can rule out. So this multi-scale coding is a genuine phenomenon. Okay, so at this point we're really perplexed. What's going on, and why would this multi-field, multi-scale representation be useful? So we joined forces together with the theoreticians, theoretical neuroscientists, Yonatan Hadef and Misha Tzodix, to try to understand what could be the function of this multi-scale representation. And we compared here 5 encoding schemes. Here on the left is kind of the classical scheme that we have in mind, that you have, let's say in this case, you have 10 neurons, for example, and this is 200 meters along the tunnel, and each neuron has one single small place field, so a 1 meter place field. So this is like what we typically think about place cells. You can also think of neurons that have single fields but are very large, so scaling up. I'll skip that. This is sort of recapitulating the Albert Lee model, but I won't talk about that. And then we have 2 multi-field schemes. One is that you have, let's say here, 10 neurons, and all the neurons have multiple fields, but each neuron has one scale. So this neuron has 3 fields, but all of them are the same. Another neuron might have many fields, but they're all kind of medium sized, and a third neuron can have a lot of fields, but they're all small in size. And we want to contrast this with what we are finding, and that is the same neuron has all possible scales. Try to understand why this would be useful. Okay, so this is what we are after, try to compare these. And so first of all, so we did a decoding approach. We did both maximum likelihood and population vector decoding. I'm just showing you the maximum likelihood results. The results are basically the same qualitatively. So this is showing the minimal number of neurons required to reach a particular decoding error of 2 meters error in this case, as a function of environment size. So we looked at environment size from 20 meters all the way to 1 kilometer. And you see that these 3 first schemes kind of shoot up. So really you need many more neurons to represent large environments, whereas these 2 schemes, even for 1 kilometer environment, you can represent it with 50 neurons, with a very small number of neurons. So this is the intuitively it's clear because it's kind of a denser coverage here. Okay, so we can say that these 3 options makes less sense in terms of the number of neurons. They require a very large number of neurons, especially these 2. Then we want to compare kind of the thing that puzzled us most in this scenario 4, having kind of multiple fields but different sizes, kind of fixed scale for the same neuron versus what we are finding in the bats. And so the intuition that Misha and Yonatan had is the following: that if in this scenario, if you have a spike, you don't know are you here or here or here or here. Okay, so you can have catastrophic decoding errors. On the other hand, in this scenario, if you have a decoding time window that's not infinitesimal but has some length, 100 millisecond or 12 millisecond, then when you're flying through the small fields, you'll have a small number of spikes. When you're flying through the medium field, you have a medium number of spikes. And when you're flying through the large field, you have a large number of spikes. So this sort of adds extra layer of information that will allow you to decode your position. And indeed, when you compare these 5 schemes in terms of decoding error, there's a bit of a busy graph, so I won't go through all of it. But what we find is that the multi-scale kind of is similar to the bat data, has the smallest mean decoding errors, and also when we look at catastrophic decoding errors, the probability of having a catastrophic decoding error, that for this purpose we define as an error that's larger than 5% of the environment size, then this multi-scale scheme has an order of magnitude lower probability than this one, and much lower than the other ones. Okay, so it's really much better in terms of decoding errors, and all of this is done with just 50 neurons. So with just 50 neurons, you can represent an environment that's 1 kilometer in size. It starts to shoot up only close to 1 kilometer. Importantly, for small environments, you see that all these lines always meet for small environments of 20 meters. So for small environments, it doesn't really matter. All these coding schemes are just fine and are doing perfectly well in small environments. So in small environments, it doesn't matter. But when you go to large environments, 200 meters, 1 kilometer, then it does matter a lot. and we suggest that this multi-scale scheme is much better. Okay, so to summarize, the representation of very large spaces, we see spatial coding of large spaces is very different from lab setups. In particular, we have many fields per neuron, often more than 10 fields in each direction. Some fields are very large, up to 30 or even 30, I think the largest was 32 meters, but also very small fields, less than 1 meter, 58 centimeters was the smallest field. Again, if you think about 10 meters per second flight speed, 58 centimeters means 58 millisecond precision. Okay, if you think the campus is a sluggish area, forget about it. It can be very precise. Yeah. A given neuron exhibits multi-scale spatial coding that the resolutions can differ up to 20-fold for the same neuron, and theoretical analysis of decoding errors explains, we think, why the multi-scale coding is better, in particular for large environments. And so I'd like to finish, and then we'll have 5 or 10 minutes for discussion. I'd like to finish with my take-home message, and I have just one for today, and that is the importance of natural behaviors versus reductionism, and the importance of having diversity in the way we study the system. So, I'm coming from a physics background, so we are always trained with a very reductionist approach in mind. If you want to understand a phenomenon, you have to just look at one variable, control all the others, and this is the way to go. Certainly in simple systems, it can be very true, but when it comes to complex systems, things are quite different. We can discuss this in the evening, also in the panel. In particular, I'm always worried that if you control and simplify too much, then we might be losing the very thing we came to understand, which is behavior, because behavior is rich, it's context dependent, it's complex. So if we control too much, we might be losing what we're after. On the other hand, of course, reductionism is very powerful. So what I'd like to suggest that there's, it's good to have reductionist experiment. We also need to have more natural experiments. We need to have both, more diversity in approaches. And from what I told you today, we haven't done really fully natural behavior, behaviors yet. Maybe this colony that we have is kind of the closest to a really natural behavior. But still, every time we go a little step towards a more natural behavior, we find something interesting. We go to two-dimensional behaviors, things look very different. Grid cells are already not lattices. Things start to look funky. We introduced a goal in the middle of the room. I didn't have time to tell you about this, but if you introduce a navigational goal in the middle of the room, then we found vectors or kind of vectorial representations of that goal. We introduced another animal, and suddenly we found there was a representation of the position of the other animal, the time for the other animal, all sorts of things for the other animal. We increase the size of the environment, all hell breaks loose. We get this multi-scale code that looks completely different than what you find in small boxes. Okay, so every time we make a step into a more naturalistic direction, things look quite different. And I'd like to argue that this is, in fact, should be done in any field in neuroscience. And it's my little plug for a book I'm writing now for MIT Press called Natural Neuroscience, where I'm trying to argue exactly this point, that basically in almost every domain of neuroscience, especially when involving behavior, it's important to consider more natural behavior. You can think of active sensing, natural stimuli, in sensory neuroscience, neurobiology of navigation. This is what I told you about today. Real-life memory research, decision-making, I think we'll hear about that today. Social neuroscience, et cetera, et cetera. So I really think that in almost every domain, it's not just a manifesto, there's really hard data to show that neurons care about whether the behavior is natural or not. Okay, so with this, I'd like to end and thank the lab members, and especially Diora Lass, staff scientist working with me. She's fantastic and has been involved in a lot of what I've showed you today, and I think I mentioned most of the people in the lab as we went along, and alumni, collaborators, and funding sources, and thank you for listening.
Beautiful talk. Can I ask you a question about the last simulation? I don't have a good intuition for what's special about Scheme 5. So the information of where the bed is in space resides in the structure of the pattern of population activity, right? So I don't understand why can't you reorganize these spike bursts such that every neuron is approximately periodic, but you still have the same interference patterns across those. That carries exactly the same.
Right. So you think I agree with you. So you think, and this was our kind of perplexion initially, that if you kind of randomly reorder fields vertically, it shouldn't matter because your kind of population at any moment any position this would be the same. So you're right, except that if you have an integration for the decoder, if you use an integration time window that is not 1 millisecond, but is 100 or 200 or a few hundred milliseconds, then you have this extra layer of information, as I explained here. That you have small number of spikes here, a medium number of spikes here, and a large number of spikes here. And I don't have it here actually, I can show you later, I have it in my laptop. We have a graph of these decoding errors versus delta t, versus time, kind of integration time windows. Of course, when you go down to integration time window of 10 milliseconds or so, it all kind of breaks down because they're disadvantaged. Then scheme 4 and 5 will be the same. But if you have an integration time window that is longer, and over a broad range of integration time window, from about 50 or 40 milliseconds up to 500, then there is this fundamental advantage. An order of magnitude, right? Because what I showed here is that this probability of catastrophic errors, there's a difference of order of magnitude, difference of 3 orders of magnitude compared to some of the other ones. Okay, this is a big difference.
So the statement is that for a suboptimal decoder of this kind, that's the best encoding.
Yes. Mm-hmm. Yeah. Yeah.
Yeah. So just more on this. So did you optimize the tuning properties in Scheme 5? So the number of place fields of each neuron, the relative sizes of them?
Okay, so what we did, we made sure that the coverage, so the area covered, is the same in Scheme 2, 4, and 5, so then it's a fair comparison. We did draw the field sizes from the empirical distribution. So our empirical distribution of field sizes is kind of a skewed one, very well fitted by either a log normal or a gamma distribution. Gamma has a nice property that it has a scale parameter that you can easily change as the environment changes. So we drew it from basically from the empirical data.
Okay, but you didn't worry about sort of fine tuning things so that.
No. So basically the tuning here is kind of is 0 or 1, right? So it's 0. Yeah, it's kind of a rectangular tuning. Yeah, we didn't. It doesn't have a shape. Yeah.
Sequences of the data?
We're looking at this now because this is a great question. How could you have, this opens a lot of questions. This multi-scale, how would you have sequences, right? Because that means every neuron should participate multiple times within a sequence and with different resolutions. I mean, your mind blows. I don't know the answer to this, but we started looking at because now that we moved, this is one of the main reasons we moved now to the 16 tetrode recording, so now we can record several tens of neurons, and we can already do real-time decoding, so kind of behavioral scale decoding very well with just 30 neurons. So I mean, here I showed you that we can decode things with 50 neurons in this kind of model, but for real data with all the noise and everything, you can decode with 2-meter precision over the 200-meter scale. So 1% precision is pretty good with just 30 neurons or 28 neurons or so. So at behavioral level, it works very, very well. And we are starting to look at sequences. I don't have an answer, but this is definitely something we're after. Yeah.
So I have 2 questions. One, do you see differences in along the axis? Because if I remember correctly, the classic idea was that along the axis of the hippocampus.
You mean the longitudinal axis?
Yeah, you get different scales,
Right.
but now you have in the same location.
Right.
You have
Right.
And the second question is, one thing I don't understand. You said the small ones don't cluster around landmarks. That means that their precision is driven by path integration.
Not necessarily, because we use light level. So let me answer the first question.
Okay.
I don't know. We haven't looked yet. We only record in dorsal. We of course are going to go for ventral, but you can. So for those of you who don't know, there is this. For many years, there's this known gradient. The hippocampus is sort of this sausage. It has a long axis, and you have the small place field here, medium place field here, and large place field there. Single place field, typically, that's the way we think about them. But you can think of it, kind of scheme 4 is sort of, you can think of this as, this would be dorsal hippocampus with the small fields, and this will be ventral hippocampus with the larger fields, right? But no, we see in the same area this multi-scale coding. So it's very different. We haven't gone ventrally, so I don't know the answer to this. And the second question is about how are the kind of the effect of landmarks. So again, I didn't show you this, but we see, kind of, we plot the size of the landmark versus position. It's kind of, it's everywhere. All sizes are everywhere, and they don't cluster. And we show this that they don't cluster around landmarks. But the light level that we use, 5 lux, is actually quite high for the bat. So they can see at any position, they can see several landmarks forward. So, you know,
So
they
it's
can,
landmark rich.
yeah, I think
environment.
yes.
It's not because I'm thinking
Right.
about these 20 kilometers.
Right.
You wouldn't see
No, no, no.
this precision in 20 kilometers because there's nothing to latch on.
I'm in the minority in the field that thinks that path integration can get you that not very far. Of course, you know, a
But
lot of people
you
believe in
would
path
expect
integration.
it to scale with the distance from the last landmark, right?
Right, right. And certainly for 20 kilometers, things like that, I mean, path integration is useless. I mean, it accumulates noises. But I think path integration is useful for kind of closing the gaps when you don't have information, short time intervals, but certainly not for kind of large distance navigation on roads. That has to be based on kind of real information, on sensor information for landmarks. So,
We'll take two more questions only.
Yeah.
So
Jan?
I have a question about the.
you and then
Oops,
you.
I'm sorry. I'm sorry.
Yeah, no, it's okay.
About the time cells.
Yeah.
It seemed to me like most of the cells fire within the first 4 seconds maybe.
Right.
Can you speculate why that might be or not?
So actually we show, again, I don't have it here, but I can show later. I have it on my computer. I mean, the time that the animal was hanging there and doing nothing was on the order of 7-8 seconds, and sort of the time cells tiled this entire interval. So whatever, I mean, we didn't create, we didn't manipulate this time interval. Whatever time interval was there, based on the behavior of the animal, the time cells tiled it. But you're right that there's this kind of, it looks like a bit of a hyperbolic. So it's not uniform. That has been shown also in rodents. So there's better tuning early on. Also, the tuning width is sharper early on. It increases. It's not exactly Weber laws. It's not exactly delta t divided by t is constant, but it's close to that. So that sort of makes sense.
And also, do you see social time cells?
So so what I showed is that we have these, so when I trigger on the landing of the other animal, this is basically, when I trigger, I have it here, it's a bit going back, but forget about it. So when I trigger on the landing of the other animal and do the same rasters, then yes, we have neurons that respond in a particular moment after the landing of the other animal. Again, I'm doing nothing, the other animal is landing, so again, you can decode, and we've done now decoding analysis, you can decode simultaneously time and place for me, and you can decode the time for the other animal, so there is representation for the other animal. And now what does it mean? I mean, speculation-wise and episodic events, etc., we can speculate what this might mean, but in any case, there's clearly, without even going into speculations, what's the function of this, there's clearly a representation of positions and times for other animals in this area that was typically thought to be, just representing myself.
Last question?
Yeah.
Okay.
In the interest of time, maybe we could switch speakers while.
Yeah.
I also have a question about these multi-scale place fields. The first one is, usually place tiles are not stable initially, they stabilize after a certain amount of time.
Right.
This is very easily imaginable in small environments. It becomes tricky to imagine that in larger environments, particularly if it's experience-based.
Right.
The second one is, if you look at small environments, do you see a different scheme? Because this is how you usually think about it in small environments, or do you see the same scheme for bats in small environments?
This is okay. The second question is a great one. I don't know. The first one is also great, but I don't have the answer to this. Is it really that the coding scheme is different? Single fields, because there were reports. Andrew Fenton increased box from 1 meter to 2 meters, then suddenly you start seeing 2 fields, 3 fields. Okay, not 20 fields, but you see beginnings of this. But all the reports are sort of single scale. So the question is, did people just not notice it and it's in their data if they reanalyze it, or it's really different? And so I think, I don't have the answer, but I think it's a fundamental one. And sorry, the first question?
Acquisition of place fields.
Acquisition, okay, we're looking at this now.